Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
High-pressure processing in food preservation. Table of contents. Introduction, principle, equipment, effect of high pressure on microorganisms, types of bacteria and HP processing conditions, working mechanism, processing parameters, uses of high-pressure processing, HPP, application of high-pressure processing, HPP, and some examples. Introduction to high-pressure processing. High-pressure processing is a novel technology used for food preservation. In this process, food is subjected to pressure ranging between 40 and 1,000 megapascals for a specific duration. The treatment time can vary from milliseconds to several minutes, depending on the requirements. Processing takes place under controlled temperatures, which can range from below 0 degrees Celsius to above 100 degrees Celsius. The purpose of high-pressure processing is to achieve microbial inactivation or to alter food attributes in order to obtain desired food characteristics. For reference, 1 megapascal equals 145.03 pounds per square inch or 10 bar. This technology is also referred to as high hydrostatic pressure or ultra-high-pressure processing. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Principle of high-pressure processing. High-pressure processing is based on the Chatelier principle, which states, whenever pressure is applied to a system in equilibrium, the system will react to counteract the effect of constraint under high pressure. Such reactions result in a decrease in volume, leading to the inactivation of microorganisms and enzymes. Another fundamental principle of HPP is isostatic processing, which states, when the food product is compressed by uniform pressure from every direction, it will return to its original shape when pressure is released. Microorganisms, chemical reactions, biochemical and enzymatic processes, as well as the functional properties of biomolecules, are significantly affected by high pressure. Equipment of high-pressure processing. Two types of compression setups for HPP according to the pressure generation module. Direct compression, high-pressure processing. Pressure is generated by moving a small diameter piston using hydraulic pressure, which reduces the volume, allows fast compression, requires a high-pressure dynamic seal between the piston and the internal vessel surface, mostly applicable to laboratory or pilot plant systems. Indirect compression, high-pressure processing, uses a high-pressure intensifier to pump the desired pressure from a reservoir into a closed high-pressure vessel, mostly applicable to commercial-scale operations. Main components of high-pressure process systems include a high-pressure vessel and its closure, pressurizing system, heating and cooling system, materials handling system, data generation system, including a microprocessor controller. Components of high-pressure process systems, collectively functioning together. Effect of high pressure on microorganisms. The sensitivity of microorganisms to high pressure varies depending on the type of microorganism, food poisoning, or food spoilage organisms, the form of the microorganism, vegetative form or spore form, Gram-positive bacteria are generally more resistant to high pressure than gram-negative bacteria, mainly due to the presence of teichoic acid in their cell walls. Bacterial spores are significantly more resistant to pressure compared to vegetative cells because spores contain calcium-rich dipicolinic acid, which provides protection. This compound helps spores resist excessive ionization under high pressure. Types of bacteria and high-pressure processing conditions. Vegetative form of bacteria, inactivated at 400 to 600 megapascals. Spore form of bacteria, require more than 1,000 megapascals for inactivation. Staphylococcus, inactivation achieved at more than 500 megapascals for over 60 minutes. Escherichia coli, shows a 6-log cycle reduction at 405 megapascals for 10 minutes. Saccharomyces cerevisiae, shows more than 6-log cycle reduction at 405 megapascals for 10 minutes. Bacillus cereus spores require more than 800 megapascals for inactivation. Vibrio parahemolyticus achieves a 6-log reduction at 200 megapascals for 20 minutes. Listeria monocytogenes, inactivated at 340 megapascals for 20 minutes. Working mechanism of high-pressure processing. Food type. The food product is packaged in a flexible container, such as a plastic pouch or bottle, and sealed. Ethylene vinyl alcohol copolymer and polyvinyl alcohol are recommended materials for food packaging. The packaged food is then loaded into the high-pressure chamber, which is filled with a pressure-transmitting fluid for pressurization. Deaeration is carried out using a pump to remove trapped air. Pressure is then generated in the system using a pump and applied to the desired level for a specific time. After treatment, the product is removed from the setup and stored properly. Processing parameters of high-pressure processing. The cost of operation depends on time of exposure, processing temperature, processing pressure. The range of processing pressure varies with water activity and the type of food product. The processing temperature is selected based on the objective of treatment. Processing temperature applications. High-pressure cold pasteurization aims to inactivate vegetative pathogenic bacteria. High-pressure assisted pasteurization and high-pressure assisted sterilization, aimed at sterilizing low-acid foods and inactivating bacterial spores. The holding time of the pressure vessel depends on both the type of food matrix and the processing temperature. Optimization of high-pressure processing is directly related to the targeted pathogenic and spoilage bacteria. Operation modes of high-pressure processing can be carried out in batch mode or semi-continuous mode for commercial applications. In batch processing, pre-packed foods are loaded into a processing vessel. In semi-continuous processing, pumpable products are used. Industrial HPP is most commonly carried out in batch processing mode. High-pressure processing system based on orientation. Horizontal high-pressure processing system. Vertical high-pressure processing system. Uses of high-pressure processing. Pathogen elimination. High-pressure processing effectively inactivates pathogenic microorganisms, making food products safer for consumption. Shelf life extension. By reducing spoilage microorganisms, High-pressure processing extends the storage life of food without compromising quality. Innovative fresh products enables the development of new, minimally processed foods with fresh-like characteristics. Increased shellfish yields help separate meat from shells more efficiently, resulting in higher yields for shellfish products. Reduced sodium products allows formulation of low-sodium foods while maintaining safety and quality. Clean label products supports the production of preservative-free and additive-free foods that meet consumer demand for natural products. Convenience provides ready-to-eat, safe, and high-quality foods with minimal processing. Application of high-pressure processing and some examples. Jams, fruit sauces, fruit jellies, yogurts, processed at 400 megapascals, 20 degrees Celsius for 10 to 30 minutes, packaged in plastic cups. Grapefruit juices, processed at 120 to 400 megapascals, 20 degrees Celsius for 2 to 20 minutes, packaged in glass bottles. Tropical fruits, processed at 50 to 200 megapascals, packaged in paper cups. Beef, processed at 100 to 250 megapascals, 20 degrees Celsius, for 30 minutes to several hours. Packaging not specified. Rice cake, processed at 400 megapascals, 45 to 70 degrees Celsius, for 10 minutes. Packaging not specified. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!